Hi again. I want to continue on with some additional environmental factors that can affect microbial growth. And again, we're trying to keep in mind the diversity out there among microbes and how different species are adapted to different environments that have different environmental factors available or that impact these organisms. All right, so we talked pretty extensively about oxygen requirements and why different microbes have different needs with regard to good old oxygen gas, O2. Microbes can also differ in terms of their temperature needs. Not all microbes thrive at human body temperature. Remember, we've already discussed how our incubators in the lab are set to 37 degrees Celsius because that's right at human body temperature. And most of the microbes that we're studying in this course love human body temperature because we're mainly interested in microbes that impact the human body. But that's not true for all microbes. Microbes out there on Earth are adapted to many different temperature environments. Why is temperature so important? Remember how we were talking about diffusion and the molecules? Molecules are constantly in a state of motion, and increased temperature increases that motion, decreased temperature decreases that motion. Well, all that movement, the random movement of the molecules and parts of molecules, when you're thinking about proteins and DNA, these big organic molecules, they have very important three-dimensional structures. Well, you got to have just the right temperature to maintain the proper 3D structure. If temperatures get too high, that wiggling molecule is going to unravel itself and it's no longer going to work. When that happens, that molecule is said to denature, like a protein which has a 3D shape like my fist here. If temperatures are too high for that protein, it's going to unravel. And when it does that, it's said to denature. On the flip side, things like lipids, like your membrane lipids, they can become too rigid at cold temperatures. If they're at proper temperatures for that particular organism, the lipids in the cell membrane are fairly fluid. If the temperatures get too cold, they become too rigid, and then the membrane's not going to function properly, not allow things to pass across when it's supposed to. DNA also, at very high temperatures, you got that double helix shape of DNA, and it will unzip, unwind. You got two strands of DNA that are bonded together, and they will unzip or unwind if temperatures are too high. Okay, now different microbes are adapted to different temperatures, which means all of their biological molecules are programmed or designed to survive and function at these different temperatures. So what we see over here on this diagram, here is a growth curve or a growth rate curve on a graph. So over here on the y-axis, this is the rate of growth of some generic organism, and over here down on the bottom, you've got temperature. All right, so if you notice, at some point, the growth rate for the organism is going to be optimal. That's the optimal growth temperature or the optimum growth temperature over here. So if you're thinking about bacteria on a petri dish, they're going to be increasing, the colonies are going to increase in size at the fastest rate if you're at the optimal growth temperature. All right, now notice as temperatures decrease for an organism, the growth rate's going to get slower and slower and slower. That's because your metabolism gets slower and slower and slower. Chemical reactions of metabolism take place more slowly at cooler temperatures. And eventually you're going to hit a minimum growth temperature. And what that means, that's the lowest temperature at which metabolism can still occur. Metabolism can still occur. So you're still getting some growth, but growth is very slow. On the flip side, if you notice on our curve, if you go higher than optimal growth temperature, your growth rate pretty rapidly drops off. And what's happening there is proteins are denaturing, DNA may begin to unwind, lipids can become too fluid at higher temperatures. They're not holding their structure in a cell membrane as well as they're supposed to. So a maximum growth temperature, now that phrase can be confusing when you first hear it because it sounds like, well, that's going to be the temperature where you have maximum growth. No, that's the optimum growth temperature. The maximum growth temperature, like you see over here on the graph, that's the highest temperature where the organism can still survive and do metabolism, and you get some growth. All right, and so these temperature ranges are going to vary between species. Different microbial species will have different optimal, minimum, and maximum growth temperatures. All right, microbes can be divided up into different categories based on the temperature ranges at which they typically grow. And I see here on my diagram some of the, looks like some of the labels came off of the PowerPoint here. So I'm going to have to draw them in. All right, first of all, let's start off here in green, this green curve that you see here. These are called your mesophiles, and they typically grow in the range of 20 degrees Celsius to 40 degrees Celsius. 37 kind of being the optimal temperature for the growth of many of those types of organisms. Meso means middle, phile to love. So these are organisms that love middle type temperatures. And of course, we tend to make humans the center of the world and the universe, and so we're going to pick our optimal temperature as the middle or normal temperature for microorganisms. All right, now down over here in blue, these are your psychrophiles, okay? And the psychrophiles, oops, circled the wrong thing. These are microbes that have an optimal growth temperature below 15 degrees Celsius, which is 15 times 2 is 30, 27, 32. That's below 59 Fahrenheit. Now, some of these actually will thrive and grow well even at refrigerator temperatures. Refrigerator temperatures are around 4 degrees Celsius. Over here on the right-hand side, you have in purple here, and take a look at this diagram in your textbook where you actually have the temperatures on the page. In purple or pink there, you've got the thermophiles, and in brown over here, these are your hyperthermophiles. Thermophiles generally grow best at temperatures above 45 Celsius, which is 90, 81, 30, that's over 114 Fahrenheit. And the hyperthermophiles are your extreme thermophiles. Some of those actually grow and thrive at temperatures over 100 degrees Celsius. 100 degrees Celsius is 212 Fahrenheit. What's the significance of 100 Celsius? That's your boiling point of water. So there are microbes that thrive in temperatures above the boiling point of water. So lots of diversity among microbes in terms of their temperature preferences. Another group I want to point out to you, psychrotrophs. So not a psychrophile, but a psychrotroph. These are organisms that prefer warmer temperatures, like human body temperatures, but they can still grow in the cold. These can be a problem with food spoilage. And unfortunately, there are a couple of pathogens that fall into that category as well. One of them, unfortunately, is Staphylococcus aureus, which causes a number of different types of infections, but it can also cause a type of food poisoning. And unfortunately, it's a psychrotroph, which means if it gets into some foods and you have those foods in your refrigerator, it's still growing pretty well in the refrigerator. Most organisms that cause human disease are mesophiles, and when you put them in the refrigerator, which is over here, about 4 degrees Celsius, notice the green line, there's your minimum growth temperature. It's way above refrigerator temperature. So most of those types of organisms aren't going to grow in a refrigerator. They're not going to be able to metabolize. They're just going to kind of be in a state of suspended animation. Your psychrotrophs, though, on the other hand, they are capable of growing in a refrigerator and can be involved in food spoilage. Let's see, I've got a YouTube video embedded here. A couple of years ago, there was a story in the news about an outbreak of a type of bacteria called Listeria monocytogenes, which causes a disease called listeriosis. If you're unfortunate enough to get to become infected with listeria, this is a type of bacteria that invades the body and spreads throughout the body and causes just kind of a general infection across the body. You can develop a high fever and so forth and feel really achy and lousy. And it is a psychotroph, so it still grows pretty well under refrigerator conditions. It's present out in the environment, especially around farms where you have farm animals, and so it can get on produce. And what happened a couple years ago was there were some cantaloupes from Colorado that were contaminated with listeria, and these cantaloupes got transported around the country in refrigerated trucks, but the bacteria were still growing on the surfaces of the cantaloupes, and people ate the cantaloupes and wound up becoming infected with listeria. And unfortunately, as I was mentioning as well, Staphylococcus aureus bacteria also can grow fairly well at cooler temperatures. All right, as with temperature, microbes can vary according to their pH requirements, and they can be divided up into different categories as well. Now, the microbes that live on or inside most parts of the human body are neutrophiles. Neutro referring to neutral, so their optimal pH ranges are close to 7. 7 is neutral on the pH scale, so generally about 6.5 to 7.5. What's the pH of your human body fluids? Hopefully you remember from A&P class, it's 7.35 to 7.45. So guess what? Lots of the microbes that live on or inside the human body are very well adapted to that pH range. There are organisms called acidophiles, so they like acid pHs below pH 7. Many fungi fall into this category. They love lower pHs. There are extreme organisms that have been discovered that will live even near pH 0, believe it or not. That's a pH where if you stuck your finger in a fluid, it would be damaged very, very, very quickly. So there are microbes that are adapted to those pH conditions and thrive in them. If you're an obligate acidophile, you must live in acid pH. There are microbes, though, that are acid tolerant, which means they don't require acidic pH, but they can tolerate it. So that includes the Heliobacter pylori bacteria that we were talking about earlier. That's a microaerophile, requires lower amounts of oxygen gas than we have in our atmosphere, and it's also acid tolerant. It doesn't have to live in acidic conditions, but it can tolerate them. It's also true for a lot of your skin bacteria. When you guys take A&P1, you learn about how the sebum, the skin oil on the surface of your skin, has a pH that's below 7. It's down in the 6 range. And a lot of microbes can't really tolerate pH 6 very well. So the lower pH on the skin helps control overgrowth. You don't have overgrowth of microbes on the skin. So the bacteria that do live on our skin, and many of those are in the Staphylococcus genus, they're somewhat acid tolerant. They can tolerate the slightly lower pHs on the surface of the skin. On the flip side, if you are an alkalinophile, these are microbes that like alkaline conditions above 7, and in some cases up to pH 11.5. So there are some extreme alkalinophiles as well. All right, osmotic pressure. What does that mean? That's referring to solute concentration. So hypertonic solutions have high osmotic pressure. And that pressure is referring to the sucking pressure, the sucking of water across the cell membrane towards where you have more solutes. So if a cell is sitting in a hypertonic solution, it is being exposed to a high osmotic pressure. Water is going to get sucked out and across the cell membrane. All right, so this also impacts microbial growth. Different microbes are adapted to different uh, solute concentrations. Okay, so if you have an obligate halophile, or your textbook is using the word osmophile as well to refer to microbes that are adapted to high solute concentrations, high osmotic pressures. An obligate halophile, halo refers to salt. There are microbes that are like sodium salt, organisms that are adapted to growing in water that has a very high sodium concentration. And you can also have facultative halophiles. Remember, uh, like facultative anaerobe, that is a organism that prefers oxygen but can grow without it. A facultative halophile doesn't need high osmotic pressure, doesn't need a hypertonic fluid, but it can tolerate those high solute or high salt conditions. Good examples, Staphylococcus bacteria. And remember in class we did the mannitol salt agar plates, and we talked about how mannitol salt agar has a very high concentration of sodium chloride salt, and Staphylococcus bacteria grow very well on this type of agar. There's a very high osmotic pressure in that agar. It's sucking water out of the cell, so most microbes can't tolerate that. Staphylococcus bacteria can because they are facultative halophiles. They have methods for getting around those super high salt conditions and surviving, even though they're being exposed to them. Okay, another environmental factor that can impact the growth of microbes, radiation tolerance. So radiation refers to anything that travels in a ray of some sort, and that includes visible light, it includes microwaves, ultraviolet light from the sun. Gamma rays are the more damaging types of radiation energy that we are really thinking about when you hear like radiation from a nuclear power plant meltdown or a nuclear bomb. That would be gamma radiation. Interestingly, even visible light is toxic to many bacteria. So these light rays, good old visible light rays that we get exposed to all the time, those go inside the cells. They go inside cells. And what they do is they hit or interact with oxygen-containing molecules and can trigger many of them to become those reactive ones that we talked about, the nasty ones that we talked about in the oxygen requirement section. And once you got these guys around, these nasty oxygen-containing compounds, they'll wind up destroying the DNA and the proteins and so forth of those microbes, and then those microbes are going to die. All right, so to get around that, have you noticed in the laboratory, for example, the incubators, do they have clear doors? Are we letting a lot of light into those incubators, or do they have dark doors? So check that out the next time you're in the lab. All right, so we've seen on your plates Petri dishes in the laboratory. You probably saw some yellow colonies, orange colonies, and so forth. And why are they making those? They're not doing that to look pretty. They're making those are called carotenoid pigments. Carotenoid, like carotene or carrot, orange color or yellow color. Those molecules, what they do, the reason they have that color is they're absorbing a lot of the visible light. And when they do that, that gives these chemical compounds this color and causes the colonies to have that color. But by doing that, it makes it less likely that that visible light is going to be toxic for the bacteria, so it helps them survive in the presence of visible light. More powerful types of radiation can be used to control microbes, things like ultraviolet light and gamma irradiation. And we'll talk about those when we get into Chapter 9. There's a link here to a video you might want to take a look at. So many microbes can't handle visible light very well that there are hospitals that are investigating the use of light technology to control the levels of microbes in hospital rooms. So like exposing, after a surgery or something, exposing the room to super, 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 super bright visible light, because many microbes can't tolerate that. And humans tolerate light very well. So it's not like using ultraviolet light or gamma or radiation or really strong chemicals, which can be toxic for humans, just like they are to the microbes. So that's kind of an interesting application of visible light to controlling microbes. And we'll talk a little bit more about that in Chapter 9. Okay, I think I'm going to stop this particular video lecture there, and then the next one we're going to talk more about biofilms. We're going to talk about the different relationships that microbes can have with each other and with other organisms, with a special emphasis on biofilms, which we've already touched on, but we're going to touch on it a little bit further here in Unit 2.